Hello everyone, let's go through how we can use GENtle to do some in silico design of the DNA sequences we're actually going to build with the Genomikon kit. So here we are in the Genomikon kit project. Let's go to the sequences section. And in this section we can see every sequence that comes with the Genomikon kit. Now each of these are currently situated next to a golden lock, which means you can't edit them. You can only view them, read them, and then, as you'll see in a moment, you can mix and match in constructs. And as we move forward, more and more people are going to be saving their sequences to this area. So just be careful that when you go ahead with this, you select each and every one of these 7 sequences with the checkboxes, and don't open anybody else's sequence because that might mess you up a little bit. So once you've clicked and checked all those boxes, let's view multiple files in GENtle. And we're going to see that pop open here. And we got 7 files now, 7 sequences that are open. And you can go ahead and view and look through each and every one of these by just clicking this button here and clicking on a sequence. So for example, green fluorescent protein, this is the sequence here. If you want a little bit more information about it, you can click the little gear here, and it gives you a little more information, and you can follow some rabbit holes that we've set up for you. And so we're going to just cancel that. You can also do some other interesting analysis using the little eye here. You can look at look at what the sequence could be translated to in different frames by clicking on these buttons, and you can look at restriction sites and do a little bit of surfing around on your own. But what we want to do for the genetic assembly is let's go click this box here and navigate to the origin of replication. And the reason we're doing this is because that's our starting point for the DNA assembly tomorrow. And once you have that active, click Tools and Designer. Now, what we can see at the very top, this is our canvas. This is where we're going to drag and drop the various parts that we have access to so that we can design our constructs for the project using the Genomikon kit. And so we're starting with the anchor, which is attached to the bead, which you can read about in the lab book. There's a bunch of information there. And then we have an origin of replication, which is the sequence that tells the cell how many times to copy the construct you're building so that it maintains through time. And we can notice there's certain annotations here, like you have an anchor, you have an X prime. X primes will connect to X's, whereas Z primes will connect to Z's. Okay, so for this next step, you need to make sure that an X is next to this X prime. And so if I want to make my bacteria glow green under fluorescent light, I'm going to use green fluorescent protein. So I simply click and drag it into this little box here, and that'll add it next to the origin of replication. If I want to go ahead and make this a pretty complicated system, I can try and also express a different one of these colored proteins after they're expressed, but you'll notice that now they start with X's and I have a Z, so I need to have a Z here, and so I can use this linker. So this linker is a short DNA sequence that basically turns the N back into an X prime. So if I go ahead and click and drag this guy, we'll see that I am following the rules, and I have a Z next to Z prime, whereafter I can now add the same. I could add the exact same construct if I wanted to try and make my bacteria glow very green. That's called gene doping. But I would like to see what it looks like if I also simultaneously express a red fluorescent protein. And I'm going to go ahead and drop that in there. So now I have an origin of replication, a green fluorescent protein cassette, a linker, a red fluorescent protein cassette, and now I need to finish this off. This is a pretty complicated system already. The more pieces you add to the end, the less likely chance you have it working properly. So we're going to go ahead and finish this with a cap. Okay, and the reason you need to have a cap at the end is because the cap actually binds to an anchor and circularizes the DNA once a year at the end of your assembly. So I'm going to go ahead and note that I have a Z prime here and I have a Z here, so I can go ahead and drag and drop this in. There we go. So now I have this fairly complicated system, okay, and this is what I want to inspect now. So I can click the blue link next to it, and that will take me back to the DNA canvas. And so now I can look at my sequence in all of its glory. You can look at the plasmid mapper here. You can see this is the origin of replication. You can drag it. Oh, now I'm floating over the fluorescent protein. Here's the linker. Here's the red fluorescent protein in the last bit here. Don't know what's happening, but that is where our cap is with our chloramphenicol resistance. And the last thing that we should do here is I'm going to change the name. So I'm going to change, just click on that. You can select all or shift down. And I'm going to call this Justin's Design. Okay, and that's good enough. I'll click off of that. If I want to, I can click on this gear and I can change this description here. Right now it's maintaining the description from the original sequence, which was the origin of replication. I can say this is Justin's sequence, which is an ORI, and then it is, I believe, a GFP, and then a linker, and then an RFP, and then a chloramphenicol, and followed by a cap. And I'll just quickly put an anchor here. And that's my sequence. I expect this to express both red and green colors. And because it's fluorescence, red and green, when the two light waves connect, it makes yellow. I anticipate this will turn the colonies yellow when excited with light. Great. I don't have to put that. I just decided to now. You can describe this however you want. It's your sequence, so I'm going to save those changes. The last thing you do is you click on Synbiota and you click Save to Synbiota. All right, you've done some serious genetic design here, and now you have this available for you to do the actual assembly kit. And when we do the kit, you'll be able to log your results in Synbiota and link it back to the sequence you've created. So now if I go back here, I'm just going to refresh. There we go, Justin's Design. So you can check out my sequence if you want to as well. I ask you not to edit it. And that's how we'll do some in silico design. Nice chat.